Good afternoon, everyone. So as Terry has mentioned, I was a member, or I am a member of the Oakley Lab for the past 2.5 years, studying the mechanisms that regulate the fundamental processes that regulate spermatogenesis. Today, I'm going to share with you one of the few projects I've been working on through my journeys in SMB. Infertility affects approximately 10% of all reproductive age couples worldwide. Male factors account for approximately 50% of the cases of infertility. Male infertility, most of the cases are idiopathic, meaning with no known causes, but some of the known causes include dysregulation of the Spermatogonia stem cell population and also side effects from cancer therapy. Some basic background information. This is a cartoon representing sperm production within the mammalian male. Seminiferous tubules can be found within the testes. And when looking at the cross-section of a seminiferous tubule, you can see various germ cell lineages and the supporting cells, such as Sertoli cells. Spermatogenesis is supported by it's a cyclic process that produces millions of sperms every day, and is supported by a population called Spermatogonia stem cells. Spermatogonia stem cell is a component of a broader population called Spermatogonia. The Spermatogonia population includes undifferentiated and differentiating population. Within the undifferentiated population are Spermatogonia stem cells and progenitors. The Spermatogonia stem cells are found within a rare subpopulation of the asingle population. They self-renew to maintain a foundational pool that gives rise to the progenitor population. The progenitor Spermatogonia then transiently amplify into a single, a pair, and A-aligned cells. That would then go through differentiation and commit to spermatogenesis. It is very important to remember that the progenitor population has A-aligned and A-pair, and those cells do not complete cytokinesis, which means they are connected by an intercellular bridge, giving them the name A-pair and A-aligned cells. Previously, we do not have a known marker for the Spermatogonia stem cell population. So therefore, it is very important to define a marker so we can further study the mechanisms that regulate the Spermatogonia stem cell pool. Although we don't have a marker for the Spermatogonia stem cell population, we do know some of the functional features for the Spermatogonia stem cells. Spermatogonia stem cells have the capacity to regenerate the progenitor Spermatogonia population following each round of differentiation, which is generating spermatogenesis, and cytotoxic elimination. And most importantly, they have to regenerate spermatogenesis following transplantation into a donor testis. If the capacity to regenerate spermatogenesis and the function of Spermatogonia stem cells are impaired, then there will be infertility, and one of the phenotype is Sertoli cell-only syndrome, meaning that the seminiferous tubules are void of germ cells. As you can see here, the various germ cell lineages. And in Sertoli cell-only syndrome, you do not see any germ cell lineages, which will cause infertility. With this in mind, the Oakley lab utilized some very unique experimental techniques to study the Spermatogonia stem cell population. The first one that I'm going to introduce to you is a transgenic mouse that has been generated from the Oakley lab. So in 2011, the Oakley lab has identified a protein that is a putative marker for the Spermatogonia stem cell population called inhibitor of DNA binding for protein, ID4. In May this year, we published another article which generated a transgenic mouse where GFP is expressed proportionately to ID4 protein. So therefore, any cells that express ID4 also express GFP. And then when you look in the seminiferous tubule, all GFP cells label putative Spermatogonia stem cells where they are in a very rare single population. And then subsequently, we made it the ID4 GFP mouse with a Rosa mouse, which the LAC-C gene is expressed in all cells. And in the presence of XGAL, those cells will turn blue. So in this subsequent ID4-GFP Rosa mouse, the putative stem cell population express GFP, and germ cells will turn blue in the presence of XGAL for further experimental uses. And then we also have another thing that we do is called primary culture of undifferentiated Spermatogonia. Why it is so important is because when you generate a single cell suspension and select for the undifferentiated population, from each testis you can get about 35,000 undifferentiated cells. And within that, about 10%, which means about 3,000 cells, are Spermatogonia stem cells. With such low cell number, it is very difficult to do mechanistic studies. So the Oatley lab can generate primary cultures of undifferentiated Spermatogonia, and we generated this from the ID4-GFP Rosa mouse. The undifferentiated cells, which encompass both Spermatogonia stem cells and progenitors, are cultured in serum-free media on fetal cell monolayer. They can be expanded from just the 35,000 to millions and millions of cells. And after expanding, they are still proportionate to the original proportion, where it is approximately 10 to 20% ID4-GFP, indicating Spermatogonia stem cells. A final technique that is very unique to our lab is the transplantation analysis. Transplantation is the definitive way to find out if a Spermatogonia stem cell or any stem cell have regenerative capacity. We can utilize the expanded Spermatogonia culture, transplant the cells into a busulfan-treated, germ cell depleted mouse recipient's testis. 2 months post-transplantation, the testes are then collected, exposed to XGAL. And as you can see here, each of these blue colonies indicating a Spermatogonia stem cell that has regenerated spermatogenesis. And if you quantify it, it is approximately 10 to 20% of the cells that were injected into the recipient has generated spermatogenesis. So just as a reminder, the experimental techniques that we can use include the transgenic mouse ID4-GFP that marks the Spermatogonia stem cell population, primary undifferentiated Spermatogonia culture, and transplantation analysis. And utilizing this, in the paper that we published in May, we expanded the undifferentiated Spermatogonia culture that is generated from ID4-GFP Spermatogonia, expanded it, and then utilizing fluorescence activated cell sorting, we separated out the ID4-GFP positive Spermatogonia population from the ID4-GFP negative progenitor population. The subsequent populations are then transplanted into recipient testis that are germ cell depleted. 2 months after the transplantation, we collected the testis and exposed the testis to XGAL. As you can see here, it is very powerful what you can see because it shows that the ID4-GFP population is enriched in Spermatogonia stem cells, and the ID4-GFP negative population did not have any regeneration, indicating the progenitors do not have regenerative capacity, and the ID4-GFP population is the Spermatogonia stem cell population with regenerative capacity. With this in mind, we again utilize this technique of fluorescence activated cell sorting to see if we can find genes that are differentially expressed within the Spermatogonia stem cell population. We expanded this undifferentiated Spermatogonia culture from ID4-GFP Rosa mouse again. Using fluorescence activated cell sorting, separated out the ID4-GFP positive Spermatogonia stem cells from the ID4-GFP negative progenitors. And then we put it through RNA sequencing. And then subsequently, utilizing bioinformatics, we were able to garner 11 genes that are upregulated in the Spermatogonia stem cell population versus 25 genes that are upregulated in the progenitor population. And it's very exciting for me to say that within the 11 genes that are upregulated within the Spermatogonia stem cell population, one gene is of particular interest to me. It is a transcription factor known as UTF1. So my big question is, what is the biological relevance of UTF1 in Spermatogonia stem cells? Let me tell you some things about UTF1. It is also known as undifferentiated transcription factor 1. UTF1 is expressed by a very rare subpopulation of undifferentiated Spermatogonia in rats. And since the publication of this in 2008, there has not been any more publication on UTF-1 within the germline. So there is definitely a gap in knowledge for the function of UTF-1 within the spermatogonial stem cell for its maintenance. So my first question is, what does UTF-1 do? Does it influence spermatogonial stem cell maintenance? Utilizing the undifferentiated spermatogonia culture of ID4 GFP rosa, I treated the cultures with either UTF-1 siRNA or control siRNA. Subsequently, I transplanted the treatment cells into recipient testes that are devoid of germ cells. 2 months post-transplantation, I collected the testes and exposed it to Excel. As you can see here, the control siRNA, you can see colonies that have regenerated spermatogenesis. And within the UTF-1 siRNA treated testes, there are less visually of colonies. And then after taking the testes apart and quantifying the colonies, you can see that in the control, there is approximately twice as much recolonization as the UTF-1 siRNA treated, which means that UTF-1 is important. It indicates that UTF-1 might have a function in spermatogonial stem cell maintenance. So in conclusion, I have indicated that UTF-1 is very important for the maintenance of the spermatogonial stem cell pool. Next, I wanted to know what regulates UTF-1's expression within spermatogonial stem cell. I looked through the literature and saw that in the literature, in embryonic stem cell, SOX2 is a transcription factor that regulates UTF-1 expression. UTF-1 expression is upregulated by SOX2 within embryonic stem cells. And then looking in other literature, I have found that SOX2 GFP cells can be found in a rare subpopulation of undifferentiated spermatogonia. So also in the same article, germ cell specific ablation of SOX2 will lead to Sertoli cell only syndrome phenotype. And so my hypothesis is that within spermatogonial stem cell, SOX2 regulates UTF-1 expression. And to test this hypothesis, I need to know if SOX2 protein is upregulated in the spermatogonial stem cell population. To do this, I again used the primary culture of ID4 GFP rosa, expanded it, utilized fluorescence activated cell sorting to separate out the GFP positive spermatogonial stem cell population from the GFP negative progenitor population. Here is a representative image of a Western blot probing for SOX2. As you can see here, in the spermatogonial stem cell population, there is an upregulation of expression of SOX2 compared to the progenitor population. And here is a quantification, and it is definitely biologically significant after 3 different biologically independent cultures. Next, knowing that SOX2 is upregulated in the ID4 GFP population, I want to know if SOX2 regulates UTF-1's expression in spermatogonial stem cells. Again, I utilized the undifferentiated spermatogonia culture. I treated the culture with either SOX2 siRNA or control siRNA. Then I have collected RNA for gene expression analysis and protein for protein expression analysis. Here, in this graph, it shows you that in light blue is SOX2 siRNA. The efficiency is about 40% efficient. And with 40% efficiency, there is about 35% efficiency in UTF-1 transcript depletion. And here's a representative image of Western blot probing for both SOX2 and UTF-1 protein. And control, you can see the bands for both SOX2 and UTF-1 protein. And in this SOX2 depleted population, you can see a decreased amount of SOX2 protein and UTF-1 protein. And here is my bar graph to quantify the Western blots. And after 3 different replication of biological replicates, it also shows that it is definitely significant in the depletion of the protein. To know if SOX2's influence of UTF1 is specific within the Spermatogonia population, I also measure gene expression of analysis on other germ cell markers. And after measuring the transcript abundance of other germ cell markers such as SOH-RH1, SOH-RH2, PLZF, and GM3, and ID4, it shows that SOX2 deficiency did not affect any of the other germ cell markers except for UTF1. So it indicates that UTF1 might have an influence of, SOX2 will have an influence of UTF1 expression within Spermatogonia stem cells. Lastly, I wanted to find out if UTF1 promoter is bound by SOX2. To do this, I utilize CHIP PCR analysis. For those who are not familiar with CHIP PCR, first I ligate the cells, cross-link the cells, so the protein cross-link to the DNA, and then I lyse the cells to make DNA fragments, and then I utilize the SOX2 chip, which will pull down SOX2 protein that's bound to the DNA, and or just normal input DNA or negative control. I design primers for the promoter region of UTF1 promoter, and then also a non-binding region for SOX2, a region that's not known to be bound by SOX2. So as you can see here is the UTF1 promoter region. In purple is the SOX2 binding site. And I italicize with an underline on my primary sequence for the SOX2 promoter binding site. Again, for my negative control, it's the exon 1 region of UTF1, which is not known to be bound by SOX2. Here is one of my representative image of my CHIP PCR analysis within Spermatogonia stem cell. As you can see here, with the SOX2 IP, there's definitely a product indicating that SOX2 do bind to the promoter region of UTF1. And here is a positive control in embryonic stem cell, which this is a known mechanism. And again, you can see SOX2 IP, there is a PCR product. So in summary, SOX2 directly regulates UTF1 by binding to the promoter region, and UTF1 expression is very important for Spermatogonia stem cells maintenance. So how would my research be important to the field? Well, all the indication from my study shows that this is the first time someone has demonstrated the function of UTF1 within the germline, and also with Spermatogonia stem cell having this mechanism and also embryonic stem cells, this indicates this might be a conserved mechanism within other stem cell types. Some future directions. My study has indicated that SOX2 binds to the promoter sites of UTF1, and UTF1 expression is very important for Spermatogonia stem cell maintenance. Both proteins are expressed in the ID4-GFP population. So I want to know if ID4-GFP influences SOX2 and UTF1 expression. But remember, ID4 is an inhibitor of DNA binding protein, so it will not regulate SOX2 directly. And so it should be inhibiting an inhibitor of SOX2. So by looking in the literature, I found that ID4 influences microRNA function expression in Spermatogonia and also other cell types. And also, very importantly, there are a few microRNA that are known to bind to the 3' UTR region of SOX2 mRNA to post-transcriptionally regulate its expression. So this will be my further study. With this, I would like to thank everyone for coming, and everyone in my lab, my committee, graduate students and staff, and the funding from NIH that has made everything possible. Thank you.
Questions for Frida?
Dr. Nielsen?
So what genes does UTF1 regulate? Have you done any kind of in silico analysis to determine whether there's something interesting there?
So what Dr. Nielsen asked is what genes does UTF1 regulate? So that's a very interesting question, and I actually have been looking into it. UTF1 is known to bind to POIS genes, which regulate the proliferation and also pluripotency of embryonic stem cells. Those are studies that has been done in embryonic stem cells. So POISE genes are genes that are both trimethylated in H3K4 and H3K27.
So I have a question about the technology when you actually put the cells back into the animal.
Mm-hmm.
So where do you put the cells and how do they get to where they're supposed to be getting to?
I think what Dr. Oatley does is put it in the efferent duct, and then we put about 10 microliter of 10,000 cells. There's a mixed population of progenitor and Spermatogonia, and then it will just migrate to the basement membrane. There is definitely, that's what we'll control, because within the same population of cells, there will be migration differences and differences in spermatogenesis.
Yeah, I was going to say, so do you know that some of the effects that you see if certain genes are upregulated or downregulated are migratory effects or digestion or maybe ability to digest through extracellular matrix?
That's a very interesting question. I think there are influences, but I don't think, that's why we use the positive control to control cells, and we do references so that we will account for those instances, and we don't actually study the migrational every time we do the transplantation analysis. Yes, Catherine.
Frida, was SOX2 one of your 11?
That's a very interesting question. So Catherine asked if SOX2 is one of my 11 genes, and as you remember in my future study, directional study, so if microRNA regulates a gene, it's post-transcriptionally, so the transcript should be there in both population. And I have also done analysis with my RNA-seq data with another platform. With less stringency, SOX2 is there, but we were very stringent in our RNA-seq utilizing the tuxedo package. So therefore, SOX2 is not there. Any other questions?
Let's thank Frida again.